Genus in the taxonomic hierarchy. Taxonomy provides a hierarchical classification system for all living organisms. The genus level occupies a critical position in this hierarchy, sitting between family and species. A genus groups closely related species that share specific derived characteristics from a common ancestor. For example, humans belong to the genus Homo in the taxonomic hierarchy. The genus level is important for several reasons. It organizes closely related species into manageable groups. It indicates evolutionary relationships between species, and it provides a practical unit for scientific classification and communication. As we can see, the genus level serves as a crucial intermediate classification between the broader family level and the more specific species level. Binomial nomenclature is the formal system of naming species of living things. This system was established by Carl Linnaeus, a Swedish botanist, in the 18th century. The scientific name in binomial nomenclature consists of two parts. The first part is the genus, followed by the species. There are specific rules for writing scientific names. The genus is always capitalized, while the species is lowercase. Both are written in italics, and together they form the complete scientific name. Let's look at some examples of binomial nomenclature. Panthera leo is the scientific name for the lion. Homo sapiens is the scientific name for humans. Quercus alba is the scientific name for the white oak tree. A single genus can contain multiple related species. For example, the genus Panthera includes several big cat species. These include Panthera leo, the lion, Panthera tigris, the tiger, Panthera onca, the jaguar, and Panthera pardus, the leopard. Binomial nomenclature provides scientists with a universal way to identify organisms precisely, transcending language barriers and cultural differences. Genus naming conventions follow strict rules in scientific taxonomy. The first rule is that genus names are always capitalized, regardless of where they appear in a sentence. Second, genus names must be italicized when printed in scientific literature or underlined when written by hand. Third, genus names are typically derived from Latin or Greek roots, often describing a characteristic feature of the organisms. These naming conventions create consistency across the taxonomic system, helping scientists communicate accurately about organisms. Let's look at some examples of properly formatted genus names. Notice how each is capitalized and would be italicized in scientific publications. To date, scientists have documented approximately 510,000 genus names across all living organisms, from bacteria to plants and animals.
Understanding monophily is essential for proper genus classification in taxonomy. Monophily comes from Greek, meaning single tribe, and refers to a specific way of grouping organisms based on their evolutionary relationships. Let's visualize an evolutionary tree to understand monophily better. A monophyletic group includes a common ancestor and all of its descendants. Here's an example of a proper monophyletic group on the left branch. In contrast, if we exclude any descendants from the group, it is no longer monophyletic. This is called a paraphyletic group, which is not suitable for genus classification. Monophily is crucial for genus classification for several reasons. First, it ensures evolutionary coherence, meaning all members share a common evolutionary history. Second, it reflects natural relationships rather than just similar appearances. Third, monophyletic groups provide predictive power about traits. If one species has a trait, related species often have similar traits. Finally, monophyletic classification helps create a stable taxonomic system that doesn't need frequent revision. A concrete example of monophily in genus classification is the Panthera genus, which includes lions, tigers, jaguars, and leopards. All these species share a single common ancestor, making Panthera a proper monophyletic group.
Genera must demonstrate clear evolutionary, ecological, or morphological differences from one another to be considered distinct. There are three main criteria that separate genera. First, evolutionary distinctness refers to significant genetic divergence. Second, morphological distinctness refers to clear physical differences. And third, ecological distinctness refers to adaptation to different niches or habitats. Let's examine some examples of closely related but distinct genera. Our first example looks at distinct felid genera. The cat family, Felidae, includes several distinct genera. Two well-known genera are Panthera, which includes big cats, and Felis, which includes smaller domestic and wild cats. Despite belonging to the same family, Panthera and Felis show clear differences. Panthera species can roar due to a modified larynx, have larger body sizes, different skull morphology, and distinct genetic markers. In contrast, Felis species can purr but cannot roar, are smaller in size, employ different hunting strategies, and have their own distinct DNA sequences. Our second example examines distinct genera within the canid family. The dog family, Canidae, includes several genera. Two prominent genera are Canis, which includes dogs, wolves, and coyotes, and Vulpes, which includes various fox species. Canis and Vulpes genera show several distinct characteristics. Canis species typically exhibit social pack behavior, have rounded pupils, larger body sizes, different chromosome counts, and distinct vocalization patterns. In contrast, Vulpes species are more solitary, have vertical slit pupils, are smaller with bushier tails, prefer different habitats, and represent a distinct genetic lineage that diverged millions of years ago. To summarize, genera are considered distinct when they show clear genetic divergence, typically representing millions of years of separate evolution. There must be consistent trait differences across all species in each genus. True genera should not have intermediate forms between them. And finally, there is often reproductive isolation between species of different genera, though exceptions can occur. A genus represents an evolutionary unit, a group of species that share a common ancestor and evolutionary history. Species within the same genus share a more recent common ancestor with each other than with species in other genera. They typically have similar genetic characteristics and exhibit related morphological traits. This simplified phylogenetic tree illustrates how a genus represents an evolutionary unit. All species trace back to a common ancestor. At this point in evolutionary history, a divergence occurred, resulting in two distinct lineages that would eventually become separate genera. Species within genus A share a more recent common ancestor with each other than with any species in genus B. This shared ancestry is reflected in their genetic and morphological similarities. In taxonomy, a genus represents a monophyletic group, all species descended from a single common ancestor. These species are more closely related to each other than to species in other genera, having diverged more recently in evolutionary history. Exploring well-known examples of genera across different kingdoms of life. Genera can be found across all major kingdoms of life, including animals, plants, fungi, and bacteria. The genus Homo in the kingdom animals includes humans, Homo sapiens. Members of this genus share characteristics like bipedal locomotion, enlarged brain capacity, complex language capabilities, and opposable thumbs. The genus Canis in the kingdom animals includes dogs, wolves, coyotes. Members of this genus share characteristics like carnivore dentition, digitigrade stance, well-developed sense of smell, and social pack behavior. The genus Felis in the kingdom animals includes domestic cats, wild cats. Members of this genus share characteristics like retractable claws, vertical pupil slits, specialized carnivore teeth, and solitary hunting pattern. The genus Quercus in the kingdom plants includes oak trees. Members of this genus share characteristics like lobed leaves, acorn fruit, monoecious flowers, and strong, dense wood. The genus Amanita in the kingdom fungi includes deathcap, fly agaric mushrooms. Members of this genus share characteristics like cap and stem morphology, universal veil-forming vulva. Many contain deadly toxins and mycorrhizal relationships. The genus Escherichia in the kingdom bacteria includes E. coli and related species. Members of this genus share characteristics like rod-shaped bacilli, gram-negative facultative anaerobes, and fermentation capabilities. These well-known genera illustrate how taxonomists group organisms with shared characteristics into the same genus category. Let's examine how the genus concept applies to animal classification. Let's examine three well-studied animal genera that illustrate how taxonomists classify animals. The genus Panthera includes large cats such as lions, tigers, jaguars, and leopards. These species share a specialized larynx structure that allows them to roar, distinguishing them from other cat genera. The genus Ursus encompasses bears like the brown bear, polar bear, and American black bear. They share plantigrade posture and non-retractile claws, among other features. The genus Corvus includes intelligent birds like ravens, crows, and jackdaws characterized by complex social behaviors and vocalizations. Taxonomists use three primary types of evidence when defining animal genera. Let's visualize how the Panthera genus fits within the taxonomic hierarchy using a branching tree diagram. Let's explore the detailed criteria that taxonomists use when defining animal genera. Morphological characteristics include skeletal structure, dentition patterns, and external features that reflect evolutionary adaptations. Behavioral traits such as social organization, reproductive behaviors, and feeding strategies often align with genus boundaries. Genetic evidence has become increasingly important, using DNA sequence homology, chromosomal structure, and molecular phylogenetics to confirm evolutionary relationships. To summarize what we've learned about animal genus classification, animal genera represent evolutionary units with shared ancestry, supported by multiple lines of evidence, including morphology, behavior, and genetics. In microbial classification, identifying genera presents unique challenges compared to plants and animals. Microbial classification faces unique challenges due to the microscopic size of these organisms, which limits morphological observations. Many microbes may look similar under a microscope, yet represent vastly different genetic lineages. Additionally, microbes evolve rapidly and can transfer genetic material horizontally between species, complicating classification. Traditional microbial classification relied primarily on observable characteristics. Microbiologists would examine morphological features, growth patterns, biochemical reactions, and cell wall composition. For example, genera like Streptococcus and Staphylococcus were differentiated by their arrangement patterns, chains versus clusters, despite both being spherical bacteria. Modern microbial classification has been revolutionized by genetic sequencing technologies. Techniques like 16S ribosomal RNA gene sequencing for bacteria and internal transcribed spacer sequencing for fungi allow precise genus determination. Whole genome sequencing and average nucleotide identity analysis provide even more accurate classification of microorganisms. By comparing specific genetic sequences, microbiologists can now determine evolutionary relationships that were invisible using traditional methods. Genetic sequencing has revolutionized microbial taxonomy, revealing evolutionary relationships that were previously unknown. Taxonomic trees based on genetic data have revealed that many traditional genera are actually not monophyletic, meaning they don't share a common ancestor. For example, major bacterial phyla like Proteobacteria contain genera such as Escherichia and Salmonella, while firmicutes include Bacillus and Staphylococcus. A notable example of reclassification is the genus Clostridium, which genetic analysis revealed to contain organisms from multiple evolutionary lineages, leading to the creation of several new genera. The impact of modern genetic classification methods on microbiology has been profound. Genetic sequencing has revealed vast microbial diversity previously unknown to science, sometimes called microbial dark matter. It has shown that similar morphologies can evolve independently, leading to convergent evolution in microbes with similar appearances but different genetic heritage. This has led to significant reclassification of many traditional genera and enabled more precise identification of pathogens in clinical settings. Comparing traditional and genetic classification methods reveals the revolutionary advances in our understanding of microbial taxonomy. Genetic sequencing has fundamentally transformed our understanding of microbial genera, revealing evolutionary relationships that were invisible to earlier scientists. The role of genus in species identification is fundamental to biological classification. When identifying an unknown organism, taxonomists navigate through the hierarchical classification system. The genus level is particularly crucial for identification. It represents a collection of closely related species that share distinctive characteristics. A single genus can contain numerous species. For example, the oak genus, Quercus, includes approximately 600 species worldwide. Let's look at a practical example of how genus characteristics help identify an unknown species. The oak genus, Quercus, has several key characteristics. These include woody growth form, spiral leaf arrangement, and their distinctive acorn fruits. Knowing the genus dramatically streamlines species identification. With genus knowledge, you can quickly narrow your search to only oak species, focusing on specific traits that differentiate them. Without genus knowledge, you would need to compare your specimen against hundreds of thousands of flowering plant species, making identification extremely challenging. Let's summarize why genus-level knowledge is so important for species identification. Genus knowledge narrows the potential species pool dramatically, provides distinctive characteristics to look for, creates a manageable identification pathway, and serves as the foundation for taxonomic keys. Genus and evolutionary relationships. Genus classification serves as a window into evolutionary history, revealing the shared ancestry of closely related species. Evolutionary trees depict the relationships between species. Organisms placed within the same genus share a recent common ancestor. For example, the genus Panthera includes lions, tigers, and jaguars, while the genus Felis includes domestic cats, wild cats, and sand cats. Species within the same genus typically share important traits due to their shared evolutionary history. These shared traits can be morphological, like body structures, ecological, such as habitat preferences, or behavioral, including social organization patterns. Let's look at a specific example, the genus Genus, which includes wolves, dogs, and coyotes. While these species have evolved different adaptations, they share numerous traits that reflect their common ancestry. Members of the Canis genus share similar skull structures, specialized carnassial teeth, pack hunting behaviors, complex social hierarchies, and similar vocalization patterns. Genus classification provides valuable insights into evolutionary history, allowing scientists to predict traits and behaviors based on phylogenetic relationships. This understanding helps researchers in fields such as comparative genomics, conservation biology, and the development of evolutionary models. Genus classification plays a crucial role in modern agriculture, guiding practices from crop development to pest management. Looking at important crop genera, we find Solanum, which includes potatoes, tomatoes, and eggplants. Similarly, the Brassica genus includes cabbage, broccoli, turnips, and canola. These related species often share cultivation requirements. Genus-based crop development leverages the genetic similarities within a genus. For example, the Triticum genus includes bread wheat, durum wheat, and spelt. These related species share important agricultural traits like growing conditions, disease resistance profiles, and milling properties, which guides breeding programs. Genus-specific pest management takes advantage of common susceptibility patterns within a genus. For example, in the citrus genus, we see shared patterns of susceptibility to diseases like citrus canker and citrus greening. Genus classification guides several practical agricultural applications. In crop rotation planning, farmers avoid planting crops from the same genus in succession to prevent disease buildup. Breeding programs often transfer beneficial traits between species within a genus as they share compatible genetics. And pest management strategies are developed at the genus level, targeting common vulnerabilities shared among related species. Understanding genus relationships helps farmers optimize yields while reducing pesticide use and breeding time. Genus classification plays a critical role in medical and pharmaceutical research, providing essential frameworks for scientific advancement. This taxonomic level is particularly valuable for three key applications: predicting bioactive compounds, understanding pathogens, and developing targeted treatments. In drug discovery, researchers leverage the predictive power of genus-level relationships. When a species produces valuable compounds, related species in the same genus often yield similar molecules. Several genera have profound importance in medicine. The genus Penicillium gave us penicillin antibiotics, while Streptomyces produces numerous antibacterial compounds, including streptomycin. Plasmodium species cause different forms of malaria. Plants like Taxus provide cancer treatments, while Digitalis contains cardiac glycosides used in heart medications. The genus Taxus, or yew trees, demonstrates how related species often produce similar bioactive compounds. Both the Pacific and European yew contain taxanes with powerful anti-cancer properties. Similarly, pathogens within the same genus often cause related diseases. Different Plasmodium species all cause malaria, though with varying severity and characteristics. Genus classification enables targeted bioprospecting in new species, helps predict disease mechanisms, and guides the development of treatments. This taxonomic level continues to be essential for streamlining pharmaceutical research and improving medical interventions. Genus classification plays a vital role in conservation biology by helping scientists identify and protect groups of related species that share similar characteristics. Conservation biologists use genus-level classification to identify groups with shared ecological requirements, develop protection strategies for related species, and prioritize conservation efforts efficiently. The umbrella species concept is a key conservation strategy. When one species in a genus is protected, other members of the same genus often benefit indirectly. For example, in big cat conservation, efforts to protect tigers in the genus Panthera also benefit other species in the same genus, like lions and jaguars. Conservation of tiger habitats benefits all Panthera species that share similar ecological requirements and face similar threats. Species within the same genus often share key ecological requirements, including habitat preferences, dietary needs, reproductive strategies, and climate tolerance. In plant conservation, protecting one species of magnolia can benefit the entire genus. Many magnolia species have similar soil, climate, and pollinator requirements. Protecting magnolia habitats preserves ecosystems for multiple endangered species in the genus, making genus-level conservation strategies particularly effective. Conservation strategies at the genus level include habitat conservation for entire genera, ex situ conservation in botanical and zoological gardens, seed banks for plant genera, and taxonomic action plans for threatened genera. Ultimately, genus-level conservation offers significant benefits. Protecting one species helps conserve genetic diversity and evolutionary potential of the entire genus, providing a more comprehensive approach to biodiversity conservation. Unlike some scientific classifications that have clear, objective boundaries, genus definitions can be surprisingly subjective. Different taxonomists may draw genus boundaries differently based on which characteristics they consider most important. For example, taxonomist A might split organisms into two separate genera based on distinctive physical features, while taxonomist B might group them into a single genus based on genetic similarities. These disagreements arise because taxonomists may prioritize different characteristics when defining genera. We can see this subjectivity in action through several well-known examples of taxonomic disagreements. These subjective decisions reflect the fundamental challenge of imposing discrete categories on the continuous process of evolution. Genus classifications are not static. They change over time as our scientific understanding evolves and new evidence emerges. Several key factors drive these reclassifications. Advanced genetic sequencing has revolutionized our understanding of evolutionary relationships. Phylogenetic analysis, morphological reassessments, and molecular clock dating also contribute to taxonomic revisions. Genetic evidence has become particularly influential in revising genus classifications. DNA analysis frequently reveals unexpected relationships between species that look similar but have different evolutionary histories. Let's look at a classic example of genus reclassification. Before the 1990s, many big cats were classified in the genus Felis along with domestic cats, Lions, for instance, were known as Felis leo. Modern genetic analysis revealed that big cats like lions, tigers, and leopards form a distinct evolutionary group. This led to their reclassification into the genus Panthera, making the lion Panthera leo. Plant classifications have also undergone significant revisions. Hostas were traditionally placed in the lily family Liliaceae. However, DNA studies revealed that hostas are actually more closely related to asparagus than to true lilies. As a result, they were moved to the family Asparagaceae, though the genus name hosta remained unchanged. Modern taxonomic revisions follow a structured scientific process that ensures accuracy and consensus. It begins with collecting new evidence, typically genetic data combined with morphological studies. The findings must be published in peer-reviewed journals and scrutinized by taxonomic authorities. Only after acceptance by the scientific community are the changes formally adopted in taxonomic databases. Many notable reclassifications have occurred across the animal kingdom. The giant panda was once classified with raccoons before genetic evidence confirmed its place among bears. Whales were dramatically reclassified when molecular evidence revealed they evolved from even-toed ungulates, placing them alongside animals like hippopotamuses. Perhaps most dramatically, modern birds are now classified as living dinosaurs, a reclassification that reflects our evolving understanding of dinosaur-bird evolutionary relationships. Overlapping characteristics between genera present one of the most significant challenges in taxonomy. In an ideal taxonomic system, each genus would have clearly defined, unique characteristics that separate it from all others. However, nature rarely conforms to such neat categories. Taxonomists often face situations where closely related genera share many characteristics, making classification challenging. Genera often share several characteristics while maintaining a few unique traits. The overlapping characteristics create blurry boundaries between closely related genera. Let's examine a specific case study where the boundaries between genera are particularly challenging. Pine trees, Pinus, and fir trees, Abies, are two conifer genera that share many characteristics and can be difficult to distinguish. Both pines and firs are evergreen cone-bearing trees with similar geographical ranges. To distinguish them, taxonomists must examine specific traits like needle arrangement, cone orientation, and cone persistence. Similarly, the butterfly genera Vanessa and Polygonia share numerous traits that make classification challenging. Despite DNA evidence suggesting they should be separate genera, these butterflies share wing patterns, feeding behaviors, and larval host plants, creating significant classification challenges. To address these boundary issues, taxonomists employ multi-trait analysis. This approach integrates multiple lines of evidence, including morphology, DNA sequences, ecological niches, behavior, biochemistry, development patterns, and geographical distribution. By examining multiple traits, taxonomists can better resolve the blurry boundaries between genera, though some cases remain challenging and subject to ongoing research and debate. For many organisms, especially those newly discovered or poorly studied, classification at the genus level can be challenging due to insufficient information. While higher taxonomic levels, like kingdom, phylum, order, and family, might be determined with confidence, genus assignment often remains uncertain for newly discovered organisms. Several factors contribute to these information gaps. Often, scientists have access to only morphological data without genetic information. Sometimes only a single specimen is available, or key diagnostic features might be missing. For extinct organisms, no living specimens can be studied at all. With incomplete information, taxonomists have several approaches. They may assign an organism to genus inserte sedis, indicating uncertain placement. They might use provisional qualifiers like CF or AF before genus names. Sometimes they place the organism in the most closely related known genus as a conservative approach, or they may simply wait for more specimens or data before making a formal classification. Let's look at some real examples. Duobrachium sparksy, a deep-sea ctenophore discovered in 2015, was only observed via ROV camera with no physical specimens collected. Dendrogramma, found in 2014, had so few preserved specimens that scientists couldn't even determine its phylum with certainty. A newly described polycate worm from 2020 is referred to as CF, Novabrachium, using the CF qualifier because only a single incomplete specimen was available for study. Genus classification for such organisms remains provisional until additional research provides more complete information. This highlights the dynamic nature of taxonomic science, where classifications evolve as our knowledge grows. The concept of genus is not applied consistently across the major taxonomic groups. The criteria for what constitutes a genus varies significantly among plants, animals, and microbes. Each field of taxonomy has developed its own approach to defining genera, leading to fundamentally different taxonomic structures. For plants, classification often relies heavily on flower structure, reproductive organs, and vegetative characteristics. Animal taxonomists typically focus on skeletal features, reproductive biology, and sometimes behavioral traits when defining genera. In microbiology, genetic similarity, metabolic pathways, and cell structure are the primary criteria, with much greater emphasis on molecular data. These different approaches create significant inconsistencies in how the genus concept is applied. In plant taxonomy, subtle differences in flower morphology may be sufficient to separate genera. For animals, mammalian genera tend to be more conservatively defined than insect genera, where there's often greater splitting. Microbial taxonomists typically require significant genetic differences, often at least 5% divergence, to establish separate genera. Several factors contribute to these inconsistencies across organism types. These include historical precedent in different fields, the accessibility of different types of data, practical utility considerations, and different research traditions among scientific communities. These inconsistencies have practical implications for taxonomy and biodiversity studies. They make cross-domain comparisons challenging, lead to taxonomic inflation in some groups, and affect genus-level biodiversity metrics used in conservation and research. Modern approaches to genus classification have dramatically transformed how scientists organize and understand biological diversity. Traditional classification methods relied primarily on morphology and subjective observations of physical traits. In contrast, modern approaches utilize molecular data and computational methods to create more objective and evolutionarily accurate classifications. DNA sequencing has become the cornerstone of modern taxonomic classification. By analyzing genetic code directly, scientists can identify precise evolutionary relationships between organisms. Computational phylogenetics uses advanced algorithms to analyze genetic data and reconstruct evolutionary relationships. These methods create statistically robust phylogenetic trees that reflect true evolutionary history. Cladistics has become fundamental to modern genus classification. This approach requires that genera represent monophyletic groups, containing all descendants of a common ancestor. Collectively, these modern approaches have transformed genus classification from a subjective art to a data-driven science, creating taxonomic systems that better reflect the true evolutionary history of life. The future of genus classification builds upon centuries of taxonomic tradition while embracing cutting-edge technologies. Future approaches to genus classification will increasingly rely on genomic data, integration of molecular and morphological evidence, computational phylogenetics, and even machine learning algorithms. Taxonomists face significant challenges, including balancing nomenclatural stability with scientific accuracy, addressing cryptic diversity revealed by molecular studies, and reconciling molecular findings with traditional classification approaches. Recent discoveries like Homo naledi demonstrate how new findings continue to refine our understanding of even well-studied genera. Emerging trends include integrative taxonomy that synthesizes multiple data sources, environmental DNA sampling for detecting cryptic diversity, advanced morphometric techniques, and collaborative global initiatives to standardize genus concepts. For instance, genomic studies in the Solanum genus are reshaping our understanding of tomato family relationships at the genus level. Despite these changes, the genus category maintains its fundamental importance as a framework for biodiversity studies, ecological research, conservation efforts, and scientific communication across disciplines. The future of genus classification envisions a dynamic, integrative concept that balances evolutionary history, practical utility, and ongoing scientific progress. As we continue to discover new species and develop new technologies, the genus concept will evolve while remaining a cornerstone of biological classification.